good afternoon to everyone, and welcome to a new series of our weekly webinar. Today's webinar opens a new cycle on controlled environment agriculture, looking at how technology can help us to produce more food with fewer resources and hopefully with lower environmental impact. And this is really what the planet needs, right? So, controlled environment systems, greenhouses, recirculating aquaculture, aquaponics, vertical farms allow us to control light, temperature, water, nutrients, and even microbiology. So, this turns all this variability into something that we can actually design and manage. So, this time, we will focus on modular integrated sustainable agriculture systems, MISAs, alongside with integrated multi-trophic aquaculture, the IMTA, examining both offshore and onshore examples. And these approaches create a balanced ecosystem where waste from one species becomes a resource for others. Fish or shrimp provides nutrients captured by algae, shellfish, plants, insects, or mushrooms, and as such, we can improve water and soil quality while at the same time we are generating multiple high value products. And here at Food for Sustainability, we explore these questions across soil, water, and food systems. Our soil health and function laboratory that has now been rebranded as Regenera Lab decodes soil ecosystems through microbiome analysis and decision tools for regenerative farming. Also, Ampli Aqua demonstrates innovative aquaponic and onshore IMTA, while the Idanha Rural Food Park connects production, processing, and entrepreneurship, all in Portugal's interior, functioning as a living lab. So, together with these infrastructures that function as a living lab, where soil, water, and food production are tested in practice. So, I really hope that you enjoy our topic today. And to help us guide us through this discussion, we have with us three speakers who bring along complementary perspectives along this water, feed, microbes market chain. So I'm very pleased to introduce you Kevin Fitzsimmons. He is a professor at the University of Arizona. He will present MISAs, modular fish, plants, insects, and mushroom system with decades of experience advancing sustainable aquaculture globally. We also have the pleasure to have Ioannis Tzovenis. He's the MicroPhykos founder, shares offshore IMTA expertise, algae biotechnology, marine hatcheries, and nutrient cycling from his more than 40 EU projects. And we are also expected to be joined by Tiago Irish. He's the Sorgal Technical Director, and he addresses onshore field solutions, aquafeed innovation, byproduct valorization, and sustainable nutrition for scaling IMTAs and MESAs systems. We know that we had a problem with Tiago. Let's see if Tiago can join. If not, we have prepared something for you. So we're going to have fun anyway. So all together, our speakers will help us explore how controlled environment technologies and IMTA can contribute to more circular, resilient, and regenerative agri-food systems, and particularly how regions as Idanha can position themselves as testbeds of these innovations. And just before we start, let me just remind you some of our house rules. So these webinars are recorded. So if you do not wish to appear, I will ask you to have your cameras off although we prefer to see your faces. It's much nicer from this end. Questions are allowed through the chat here or through the chat in Slido. You know that I will direct your questions to our speakers at the end of their presentations. And of course, as usual, we will award you a certificate at the end of the webinar. You just need to complete a very short, easy peasy questionnaire. And I'm sure you also have fun while you go through the questions that we have prepared for you. Okay? So we are about to start, but let's all together do a short warm up. Let's turn to Slido to see what the questions that the team has prepared. So I invite you all to jump on Slido to start testing our knowledge about controlled environment agriculture. You know, this is very easy to do. You can either scan the QR codes and in your mobile you access to Slido, or you can use a browser, the browser in your PC. Just tap in www.slido.com on the hashtag box. Very easy. F4S Academy. Let's start. Yeah, you are all invited to join Slido. Okay. So first question. According to FAO, what percentage of aquatic animals for human consumption came from aquaculture in 2022? And for the percentage, we can see this is already a fairly high number, but exactly which number, let's see. So FAO published in 2024 the State of World Fisheries and Aquaculture, and has shown that aquaculture has reached an historical milestone. For the first time, the farming of aquatic organisms, which includes fish, shellfish, and aquatic plants, has become the main source of aquatic animals for human consumption. So in 2022, the global aquaculture production reached 113 million tons, while capture fisheries captured fisheries have remained quite stable at around 92 million tons per year. So, and this is, as we know, largely because the wild stocks are already very close to their sustainable limits. So, taken together, this means that aquaculture now accounts for 57% of all aquatic animals produced for food. I'm sure this percentage is going to increase in the near future. You're absolutely right. But at the moment it's 54%. And this shift highlights that the growing importance of aquaculture in ensuring global food security. And it also reinforces the need to develop more sustainable and resource efficient production systems, such as circular aquaculture and nutrient recycling approaches. Thank you so much for taking part. Let's learn about another topic. This question now is for RAS systems. So, do you have an idea how much less water can a recirculating aquaculture system, so the RAS, use compared to traditional pond aquaculture? So, are we addressing savings of 20%, 60%, 80%, or do we have savings of more than 90%? So while you vote, just a little bit background for you. So recirculating aquaculture systems, or the RAS, are designated to dramatically reduce water use. And why? By continuously treating and reusing the same water inside the system. Instead of relying on large volumes of new water, RAS uses several treatment steps. For example, mechanical filters remove solid waste such as the fish feces and uneaten foods. Biological filters host nitrifying bacteria that convert toxic ammonia into less harmful nitrates. So additional components remove carbon dioxide, they add oxygen, and stabilize water chemistry. So we have the idea that this means almost 80% of savings, but actually evidence shows that we can go as high as savings of almost 100%. So we are within the region of 19 to 99% less water that is used in these systems. So that's really dramatic savings in terms of water, which in countries like Portugal, it's really what we need is to save the few water that we have. Thank you very much. Thanks again for taking part. And now microalgae, microalgae and proteins. So do you have an idea what is the percentage of proteins in microalgae by their dry weights? Are we talking about 20%, up to 40% of protein content? We will the range between 40 to 60%, or is it higher? We are talking about 7 to 80% of protein in microalgae. So while you vote, and we can see that this is a very important feature of microalgae. And microalgae are microscopic photosynthetic organisms that are capable of accumulating very high levels of nutrients, particular proteins. These values are comparable to major feed ingredients in aquaculture, such as, for example, soybean meal. And in addition to proteins, microalgae also provide essential amino acids, omega-3 fatty acids, vitamins, pigments, and antioxidants, making microalgae a promising sustainable ingredient for aquaculture feeds. And because microalgae can be cultivated using sunlight, nutrients and carbon dioxide, they are increasingly being explored as part of circular aquaculture systems, where nutrients for fish production can potentially be reused to grow new biomass. I'm getting to the answer. Um, so, depending on the species, we need to account that the species also has their content of protein in variable rates. But many microalgae contain between 40 and 60% of protein by dry weight. So well done. And we are talking about, for example, the Chlorella vulgarias that normally reach between 50 to 60%, while species such as the Nanochloropsis or the Tetraselmis have a range between 40 and 55%. So well done. You got this right. Thank you so much. So we do have a little bit more knowledge about what is going to happen in the session today. And I can see that Kevin is already here to tell us about MISA systems. So, Kevin, whenever you're ready, the floor or the screen is yours. Thank you.
Okay, hopefully everyone can see that okay.
Yes, we can.
So, obviously, University of Arizona is in a desert, and so a lot of our crops are irrigated, and we move lots of water around this state in big irrigation systems. And as the state has grown economically, we have 7, 8 million people here in this state now. We know that we need to really maximize what we get out of every drop of water we use in agriculture. And so at the same time, we need to reduce the environmental impacts of that agriculture in the desert. Having worked in Asia quite a bit, I was familiar with a lot of the integrated aquaculture, agriculture that happens in Asia and wanted to bring that back to the US. So starting almost 50 years ago, I began with a lot of different trials with my team, with my grad students, of growing fish, shrimp in the desert, how to integrate that with field crop agriculture and vegetable agriculture. And then as our controlled environment agriculture, greenhouse industry grew, we started doing more and more in greenhouses as well. And I think everyone here is probably familiar with aquaponics, and the simple description is that it's a hybrid of aquaculture and hydroponics, which, yeah, that's actually true, but overly simplistic. The microbial community is really the most important part of the entire system. You've got to have a diverse community of microbes in there. Not only do we have nitrifying bacteria, but we've got heterotrophic bacteria, rhizobial bacteria, oxidizing, reducing, a very broad, diverse community of microbes, including algae, bacteria, and fungi. So this is a little more realistic description of what's going on there, which I like to use in my classes to explain in a little more detail exactly what's going on in aquaponic systems. Some of the places that we applied some of this research included the Epcot Center, the land pavilion there, which is a boat ride through a series of greenhouses looking at the development of agriculture historically, and then looking at a variety of the most important crops around the world, and then how we're going to produce food in the future, with hydroponics and aquaculture being two of the most important there. And then one of the nice things about that is that we have about 30,000 people a day who go through this exhibit, if you will, learning about these things. And then there's a restaurant, rotating restaurant above that looks down into the greenhouses where they serve all of the produce and fish that are grown there within the land pavilion. Another important program that we were involved with is the Biosphere 2 project. This is built just outside of Tucson. Basically, pretty complex 1 hectare greenhouse that's completely sealed. Stainless steel floor, bonded to glass and aluminum frame structure that covers the entire thing. This had 8 people who were living inside for 2 years, recycling everything, their air, their water, their food, all their waste within the structure. So as we were doing these projects, we started looking at aquaponics as being a very practical, intensive food production system that would be able to recirculate and recycle a lot of nutrients for human consumption and for some of the other animals that were going to be grown inside of the Biosphere 2 structure. And so we started with, back in the early 80s, this was very simple aquaponic systems, some in gravel, some in nutrient film techniques, just with water flowing past the roots of the plants, dropping back down into the fish culture section with a gravel bed biofilter there. And these actually allowed us to grow a tremendous amount of produce in a very small area. We also did some sand culture, which was going to be a little more of what we were going to be doing inside of the Biosphere 2 structure, with different densities of fish, going to irrigate hundreds of plots of barley in this case. And we also did more tray culture using some rock wool and more traditional, what was becoming then traditional hydroponic production. And then we also did quite a bit of deep bed production with a variety of different crops. I didn't do this one, but I saw it in another location, which I thought was really ingenious, of just doing aquaponics in chopped down banana trunks after the bananas had been removed. As people probably know, bananas only flower once and harvest the fruit, and then the trunk normally is just chopped and allowed to decay in sight. These folks just built a frame and came up with this ingenious aquaponic type system here. So we did a lot with fish and lettuce systems. One square meter plots here that we could do multiple replications, and different growing systems, mostly with tilapia, but sometimes other fish as well, koi, catfish, bass, in these systems. We've done a lot with different types of lettuce over time, which I think is fairly standard these days. And this is where a lot of my students did a lot of their practical work on theses and dissertations. And we've also, as it turned out, found many schools, high schools, elementary schools, middle schools, that love these for education purposes. A simple aquaponic system in a high school. Most recently, I started working with an entomologist on campus who's very interested in growing insects for human and animal feeds. So we started trying to use insects in place of fish meal and fish oil in our diets, and also had a student who was interested in adding filter feeders like mussels, bivalves, into the system, not only as an additional biofiltration, but as an edible crop that could be harvested from the system as well. And so we were farming insects, turning those into feeds, which were dried. We could feed the tilapia. Water would pass to the mussel tank, into the biofilter, out into our plant bed, and pump the water back to the insect-fed fish. Had another faculty member and PhD student working with mushrooms nearby, and his interest was to take our aquaculture effluent, use that to soak, hydrate the substrate in which the mushrooms were being grown, typically in these kind of bags. We actually did this in a 40-foot container for using our myco production here. So in working with the mycologist, the entomologist, the hydroponics people, and the aquaculture people, we got talking and noticed how much we could cycle amongst these four production systems. That we could take water from the fish, our fish effluent, use that to hydrate the mushrooms. We could use that to irrigate, fertilize our vegetable crop, and that some of the wastes from the mushrooms, the plants, the fish could be used to feed insects. The insects could then be used to go into the fish feed, along with some of the other spent material from the mushrooms could also be ground up and used into the diet. And so we finally added to that a solar drying tower here in the center, which takes waste from the student union, leftover vegetable waste, other solid waste here that we can dry in the tower and then use that not only as substrate for the mushrooms, but also as feed for the insects. So this is our primary input coming here, but then within the system, we're recycling a lot here. And as we got to thinking even more about it, we could take the CO2 evolved from insects, CO2 evolved from the fish, CO2 evolved from the mushrooms, pump that into a chamber with the plants, then take that enriched, oxygen-rich atmosphere and pump it back to the insects, the fish, and the mushrooms. So we've called this a modular, integrated, sustainable agricultural system. And what we've done is started out, we got 4 walk-in coolers. These are probably something a little less than 20 cubic meters each. One for mushrooms, one for plants, one for fish, one for insects. And what we've done is plumbed these together so that we can take atmosphere from one and move within the units, and plumbing so we can move water within the units. Now, some of the larger material, we're just going to have to open the doors and walk back and forth, moving some of the material. But as far as water and air mixtures, I guess I should more properly say, we could move between here with pumps and blowers between the units. We've recently added the computer interface data acquisition system, computer down here, where we can take temperature and dissolved gases hooked up to a gas chromatograph, so we can monitor what's going on in these systems. And so this is actually operating now. We're just collecting data. We don't have any papers generated yet, but we're looking forward within the next semester or two of students having enough data to actually write up some papers. So the next step on this is we're hoping to get 4 container integrated, integrated transport containers, 40-foot containers that go on ships and trucks and trains that would be refrigerated and move into a larger unit. So, whoops. So again, just a version of what we're going on there. And then finally, it's amazing to me how aquaponic systems are popping up all around the world. Major urban centers are getting into aquaponics. We'd love to see eventually that we might increase that to more controlled environment agriculture and maybe eventually MICES systems. So I'll stop there and stop sharing and turn it back to Claudia and Anna and the team there. And it's a pleasure to present this and share this with everybody.
Thank you,
Thank
Kevin.
you.
Thanks for sharing. It was very interesting to see your system working. One thing that I note is that you need to have very healthy students because what they eat, it's the input
Yes,
of
that
your
is true.
system. So we need to make sure that they eat fresh vegetables from the best quality. So great. So
Exactly right.
Let me just address the audience. I don't see any questions. I don't know if you have problems with this chat, but we have the chat on Slido open if you want to address your questions to the speakers. And I will start, Kevin, with a clarifying question about the way your system works. Could you just highlight again the key operational difference between your MISA systems and the traditional recirculating setups? But specifically, if you can, touch on water. How do you optimize water quality, energy use, and particularly of interest is the nutrient recycling, because I can see from the participants that we have some farmers in the audience, and to see, how is this operation efficiency in terms of water quality, energy, and
Certainly, yeah,
nutrients.
great question. And, I got into aquaponics in the first place because doing fish culture out at a farm, putting the effluent on the field, it wasn't really so obvious that the plants were getting benefit from the nutrients coming from the fish. Doing an aquaponic system, and especially when the roots are just hanging in the water, it's obvious there's no soil, that all the nutrition with these plants growing very well just on the waste from the fish was much more obvious to the farmers or to students, whomever came into the greenhouse, that there was enormous nutrient value in this fish waste, and that just traditional aquaculture, throwing it to the environment was really a waste of super good nutrients. So that's why we got into it. And as we've gone on, we've noticed one of the things was that working with a lot of hydroponic producers, they said, "Oh, your fish nutrients are way too low. Your nitrates need to be, 200-300 part per million. Your phosphates need to be this. Your iron needs to be this." Luckily, our plants did not read their books. The plants grew quite well, and we attribute a lot of that to the microbial community. When you have these microbes that are converting nutrients, many times they're into forms that the plants can use more efficiently. We've got a lot of micronutrients in there. We've got rhizobial bacteria there. And I think that's one of the things that this integration allows us to be more efficient with nutrient transfer than the more traditional chemical fertilization of hydroponics.
And that's definitely a very important point to come across. It's because we need to get away from these linear systems and to ensure that we recover everything that we are throwing away. Kevin, another question that relates to the specific conditions that you have. You are in a desert and you have a
Yes.
greenhouse. Can you comment on the temperature management system that you have, particularly during the summer?
Yeah, so we use basically an evaporative cooling system, what we call a wet wall. So it's either wood shavings or a cardboard type arrangement. It's called cell deck, which actually was invented, I think, in Sweden. Munters Corporation that brought it to the U.S. And so there's just water trickling down this wall. There's a fan at one end of the greenhouse sucking air from the outside. It passes over this wet material, evaporative cooling. It is more energy intensive in that you're running fans, especially in the middle of the day. But then in the evening, it holds the temperature pretty well inside the greenhouse. So we're only cooling for several hours a day. So it is energy intensive compared to other systems, but not like compressed air conditioning.
Okay. Okay. So we do. Uh, it's demanding, but at least it's not as demanding as other systems. Thanks for that
Right.
clarification. One final question, Kevin. Can you tell us which parameters do you assess other than the yield and the physical chemical properties? What nutrients have you been assessing?
So we've been looking at the different nitrogen, ammonia, nitrite, nitrate, phosphates, organic phosphate, inorganic phosphate, obviously pH, temperature, the humidity is monitored inside, PAR, photosynthetic active radiation, in the plant chambers. Those are the primary things that we're looking at. We've been doing some environmental DNA and microbial community
Microbiome.
definitions in microbiomes. Yeah, exactly.
Okay. Great, Kevin. That's thanks for your explanations. We need to move on. So what I'm going to ask is for Sophia to direct the few questions that we still have on Slido to Kevin, and Sophia will then pop the answers here in the chat. Okay, thank you. We already have Ioannis Tzovenis on board. Ioannis, good afternoon. So now is your turn. When you're ready, please share your screen.
Yeah. Hi. I'm just sharing. Is it okay?
Yes, that's absolutely fine. We can see perfectly.
Okay, super. I'm going to talk about marine water now, okay, which is more challenging, especially for infrastructure. But we did a lot of work on land-based IMTA using sea bass hatchery effluent to grow seaweeds in a Mediterranean aqua farm, and this is in Kefalonia, in an island in the Ionian Sea, in the Mediterranean, which is one of the major, of the largest aqua farms in Mediterranean and in European space, let's say. And my name is Ioannis Tzovenis. I come from Greece. I'm working with aquaculture and algae, especially aquaculture and biotechnology, since 1983 that I started with spirulina. But I am working with all different types and species of algae. So I would like to show some, oops, I'm trying to, yeah, okay. So I'm going to describe a little bit these marine IMTA systems. So IMTA stands for Integrated Multi-Trophic Aquaculture. So we have three components. One is aquaculture, especially what we call fed aquaculture. We have different species that we feed, and especially fishes. And then we use it in a multi-trophic mode, meaning that we have different trophic levels, meaning that we have species like predators or species like detrivores or species that they are filter feeders, etc. So they have different trophic habits and they are in the same space. That's why we call it integrated. So this is what we call integrated multi-trophic aquaculture, everything in one space. Actually, in Western world, we are more accustomed with monocultures, and these kind of systems in a traditional mode, they are working in Far East for centuries nowadays, and we are trying to nowadays to simulate them. So in order to give you a very good example, if you see the left part of the screen, this is how they are working in Ireland in the seaweed company. So there is a farm with fish, and in this case salmon. There is another farm next to it with mussels and oysters that they are filter feeding on particles and on phytoplankton that is thriving on the ammonia and other waste that is decomposed by the bacteria in the area. And together, all these excretions and emissions of nitrates, ammonia, phosphorus, etc., they are taken up by Alaria esculenta, which is a big, which is a large kelp, a brown alga that is situated next to it. So all these three different components, they are working together, and I will explain what is the outcome later. The extractive species that we use, because the fed species are the fish, and the extractive species, they can be bivalves, that they are filter feeders. They can be seaweeds, okay, that they remove nitrogen and phosphorus from the fish, and they also do carbon sequestration. And under the sea cages, we can have on the bottom or in trays, we can have sea cucumber, which are holothurians, they are echinoderms, that they are deposit feeders. They eat all the fishes and everything that it is unfed or whatever is sinking down on the bottom. And this way they reduce organic loading from the fish and the mussels, because also the mussels, they are producing fishes and pseudofishes. And also another component could be lobsters. You can add lobsters. Normally they are tomatophags. They are eating dead material, but they can thrive underneath these cages, and we can, because they are very difficult in cultivation, they can be, let's say, a very good case under these cages. And after that, they can either be released to natural habitats for restocking purposes, or they can be, of course, harvested for food. Now, we have to understand that this IMTA is a transition from circular farming to from linear models to circular farming, using the whole ecosystem approach, which is in the right part. You can see what we call make, use, reuse, remake, and recycle in an continuous circle like that. But also we have to understand that all these, they are built in together within the environment, because within the environment we have the society and the economy that comes out of the society. So we have three sustainability dimensions in this IMTA. And to talk about the benefits, we're going to talk about environmental, economic, and social benefits. The environmental benefits are pretty obvious, to say the least, because we have a simulation of the natural cycle in the ecosystem. And this way, water can be cleaned up. And also, if we have a system which is onshore, then it can be discharged safely. It could be even pristine. It can be fully cleaned up and be reused. So we can minimize this way aquaculture's environmental impact, because there is such, not very big, but there is. There is a high potential for nutrient load reduction, and especially recycling, because most of these nutrients, we have to realize that they are lost in the environment if they are not reused. And bear in mind that the ocean-based protein is substantially less carbon intensive than land-based protein. So this means that we can have a diet shift to lower carbon protein that helps reducing the emissions. And also we have to realize that polyculture systems provide a variety of species that they simulate more or less what is in real terms in the environment, which enhances biodiversity. And these kind of systems, they give shelter and habitat for various other marine species. So we have a full ecosystem approach in this kind of setups. The economic benefits: alternative income for the farmer, diversification of the product. We can reduce the costly water treatment in the land-based systems using seaweeds as a filter. We have also the ability to sell aquaculture products at a premium price because there is people that they are willing to pay more for this kind of circular and environmentally sustainable products. We can have also cooperation among farmers, one farm for fish, one farm for shellfish, one farm for seaweeds, working together. This also enhances the business resilience in many aspects, environmental, global warming, economic aspects, consumer demand, etc. And don't forget that we optimize space this way. We have multiple products produced within the same space. Now, the social benefits, if we compare land farming workers to aquaculture, we see that the people in aquaculture, they have a certainty of working location. They don't have to move from village to village with different crops, etc. Because it requires high skills, they have to access to education, to health provision and appropriate housing, because normally aquaculture is taken up by corporates, big companies. We have regularity of working hours because they are working in shifts. More predictable returns in both time and value, which makes easier to have informed financial planning decisions and investments by the workers and the farmers. And finally, we have to realize that IMTA is more resilient. It's more resilient against disease outbreaks, food safety recalls, for instance, toxic blooms, and natural disasters. Always something is left over there. So there are a lot of benefits, but there are also challenges. There are challenges regarding regulation, license systems, etc. There is an uncertainty of market because you have different farm species that they are less expensive than the fees, etc. So this makes it a little bit unstable. There is a complex system that needs a very high-skilled management, and this asks for people that they are familiar and they have skills, necessary skills for this kind of operations. Okay, there are challenges. Don't fool ourselves. Now, I'm going to present you what we do in Kefalonia fisheries, and I'm going to also take the opportunity to advertise a little bit the European project that we are working with nowadays in this context, which is Nova Foodies. And what do? We use the effluent from the fish hatchery to grow seaweed. We grow ulva and Chondrocanthes td, a green and a red one. And because we are based on waste to raise these crops, we are not very sure because of the regulation that we can use them for human use, food, feed, etc. So we are directing our focus on biofertilizers and biostimulants, that they are very good also alternatives. But if we find a way to use ulva, for instance, for food, you can see here prices that they are in the European market nowadays. That makes it quite attractive and important as an additional income for the farm and any farm that might take the chances on it. I have a big picture here connecting all the parts of the system. So we have I hope you see my cursor. We have the larval units that they are in some sheds like that. We have several like that. It's a very big farm. We have the larval unit, the live food for the larval units, which is Artemia, rotifers, and phytoplankton. We have the large pre-growing unit because the larvae, they start from millimeters, and in the pre-growing unit, they become from 3 grams to 20 grams. So they grow in size. and also the amount of water that they use is substantial. Don't forget that these farms are flow-through systems. They constantly pump up water from the sea. This water is cleaned up and it passes through all these components and then has to go back to the sea. But before to go to the sea, we track all this, we duct all this water into a wastewater treatment basin, where it has 3 different steps where it gets mineralized, or the organic load, and it ends up in the sedimentation pond before it gets discharged. It is from there that we get water for our pilot plant with seaweeds here, which is in tanks. We have many tanks like that, that they get constant this water from the sedimentation tank. And there we farm the seaweeds that they are starting as seedlings in indoor facility that we have in the farm. Here you see this Hodercanthus, and here is the ulva. It's 3 different species of ulva. We have ulva lacinulata, ulva intestinalis, and ulva I don't remember the third species. So here is how they are growing into the different tanks. They become very dense, and then we crop them. We dry them in the greenhouse, and we store them for a later use by our industrial partners. So having presented all this is a result table from February to November of 25. You can see that starting from February to March, we had salinities that they were more or less stable, but we had different amounts of loads from dissolved. This is for dissolved inorganic nitrogen, this phosphate, because of the different stages of the fish farming in the hatchery. And this is how the light gets higher and higher as we approach summer. Temperature is a bit low in the beginning, but it gets higher. All these are morning temperatures. pH are stable because all this is in the seaweed tanks, and dissolved oxygen is high because they produce oxygen. So what we saw, very high yields during from May to July, and quite decent yields in the other months. And this is substantiating our hypothesis that we can have a real business working with effluent. And to tell you the truth, because we compare that with monocultures that we used defined media, we saw that the water from the farm, from the hatchery, is somehow supporting higher yields than the monoculture, which is a very, very important finding. This is a range of different seaweeds that we used. It's not only ulva and Hodercanthus. We used other reds like Spherococcus, Corympholius, and Laurentia. But also brown species like Dictyopteris membranacea and Cystoseria barbata. Now, I'm going to present you something totally different. This is our recent project with spirulina and aquaponics. This is in Uzbekistan, where we have an aquaponic system pretty much like the one that Professor Kevin Fitzsimmons showed us before, working with fish and lettuce or other salads. And from there, we get discharge from the RAS system, because there is always a discharge from the RAS. We get this discharge, and with that water, we are working with the spirulina. We are feeding, let's say, the spirulina, and then we collect the spirulina, we dry the spirulina, and although we have the ability to make human food or supplement products, we are focusing, or we are targeting fish feed development, especially carp feed development, that this carp feed is fed directly to the carp that is reared in the aquaponic system. So we are closing the loop this way. And this is a project that is funded by ICBA, which is an institute in Dubai. This is pretty much what I wanted to show you, and this is me and a few details about me. I'm happy to be here, and if there are questions, I will be glad to answer them.
We have a couple of questions. Ioannis, thank you so much for your presentation. It was very nice. And also with that beautiful picture in Catalonia, just we pay even more attention to what you are saying. Thank you so much. We have a couple of questions here in the chat. I will just pick them. There were a couple of questions about the microalgae. Do you have work on microalgae? And if so, the results that you have just shown before, can they be applied to microalgae as well? You mentioned seaweeds and microalgae.
Microalgae and macroalgae are different worlds. Okay, they have the same biochemistry, to more or less, but they are very different in how you can handle them and how they behave in this kind of systems. We have done work as, first of all, I showed you a slide with spirulina. Spirulina is microalgae. Okay, it's filamentous cyanobacterium, so it's really a small thing. The problem with this kind of RAS systems and aquaponics is that if you use microalgae, then they go everywhere. They don't stay in one place. They go with the water everywhere. So it's one problem is this. But we did several projects. We had a project with bioflocks using microalgae, chlorella in particular, and we had published also on that. So this is a way we had tilapia, and then the water was going to bioflocks. I don't know how familiar we are with bioflocks. Bioflocks are let's say, organic carbon-induced systems that they are working also with algae in symbiosis, and they recycle and they enrich and they go looping around. But in order to loop these things, we had to separate the algae from the water, so because otherwise they were in the fish, so in the fish tanks. So it was a bit difficult this way. Normally, what you can do is to have a RAS system and take the flushing, the discharge of the RAS, and work with microalgae. This is the safest way to do, and this is how they are working all over Europe that I know of.
Okay, and this is actually what we do in Ampliaqua. Thank you so much, Ioannis. One final question, and then I will ask you to direct the questions also from the chat. There is an interesting question here. It relates climate change. So, because we are, witnessing climate change, have you come across to changes in nutrition needs of species because of the change in the environment, high temperatures, dry periods, and could these systems be a solution for that?
Listen, in nature, the climate change is more, let's say, affecting our perceptions and not nature is adapting to the change. We have to adapt to the changes. That's the problem.
Yeah, the planet will keep
Be,
going. The problem is
yeah,
that,
because we
for
learn
sure.
to work in a way, and then nature comes and changes plans, and then we have to repurpose or reschedule what we're doing. But the for mussels, yeah, there is a problem because of climate change is raising the temperature in the water, and we have problems in the Mediterranean with this thing. Many mussel farms are suffering in the summertime, and now we are thinking of replacing mussels with other more resilient species that they are migrating from the Red Sea. We have Fluxopectin or pink tata that come from the Red Sea, and they are pretty much accustomed to the higher temperature, and that's it. Ulva is not really affected, but other species, we are having alternating crops somehow because these reds, they don't feel very well during the summertime when the temperature rises, but ulva goes very well. So during winter time, that ulva doesn't feel very well. We have the reds, so we can do it like that. But in the sea, where you cannot manipulate anything, this might be a problem to change crops. I'm talking about having long lines in the sea next to the sea cages. But this is something I am in close contact with the Hellenic Marine Research Institute here in Greece, that they are researching this topic exactly, how this climate change is affecting the species, and we need to simulate because you have to do research before you go and do technology in the farm. We have to learn about these changes first.
Yes, this is probably one of the reasons why we are going to see more and more food coming out from these systems, because nature is really being affected by a lot of things, namely climate change. So I'm sure that the figures that I've shown in the beginning are going to increase, and biotechnology, these systems are definitely will go to play a very important role. Ioannis, I will now ask you to kindly direct to the chat to answer the questions. Sophia has
Mhm.
already addressed the questions, so she will be popping up if she can the questions in the chat for you, because now I will call the next speaker to the stage, which is actually me and Sophia. We have something to share with you. We would like to show you an opportunity to democratize all the knowledge that we have been witnessing here. Is to invite you all to visit Ampli Aqua, and namely we wanted to introduce you a project that we designed specially for this knowledge transfer and for this democratization. So Sophia, whenever you're ready, don't forget that we have a video, so we need to share with the sound also so that our audience can follow us. Okay, we can see presentation mode, so we're ready to start. So after hearing this, our experts talking about the incredible potential of IMTA and the MISA systems, let me now show you how we are making this innovation accessible to everyone. So Amplicentro is our way of democratizing onshore IMTA systems through Ampliaqua. So, and here we turn it into a living lab where farmers, entrepreneurs, researchers, and communities at large can learn, test, collaborate, and scale the systems together. So let me start by sharing some of the specific objectives that guide this project, the Amplicentro. Centro. So what we are trying to achieve with Amplicentro. So our main goal is to take everything that we have learned from the Ampliaqua, all its technologies and knowledge, and turn it into practical, scientific, and commercial solutions that help transition to more sustainable food production. And specifically, we are focused on three big areas. So increasing sustainability in agriculture. We use hydroponic modules where fish waste feeds the plants, and we are able to cut chemical fertilizers by 19%, while at the same time producing vegetables 30% faster than soil. Secondly, we look for efficient and sustainable fish production, and through our RAS fish tanks where Ampliaqua recycles 98% of water, and we use insect protein feed, reducing, of course, environmental impact while at the same time maintaining high yields. And the third pillar is to use circular economy technology. So our microalgae bioreactor captures CO2 and nutrients from the fish water to grow spirulina that has up to 60% of dry weight in protein, which becomes fish feed or food for humans. So everything here, every single mode at Ampliaqua becomes a real world demo for these solutions. So farmers can see, can test, and can adapt to them directly. So let me just show you how we have structured this to make this happen. So this is how Amplicentro unfolds in 4 key stages: planning, knowledge transfer activities, communication, and impact. But let's be honest about it. At the heart of this project for you, of course, our audience, is the knowledge transfer activities. This is where real action happens. So to this end, we are organizing online webinars like this very cycle that you're just watching now. We also have open days at Ampliaqua where you can see the system running live. We also have hands-on workshops and RAS capacitation programs. And we have fairs and show cooking events. Yes, where you can taste our delicious pesto or try a great pizza topped with basil or rockets straight from our hydroponic towers. So these are moments where theory becomes practice, and I definitely want you to see you there, hands-on in the tanks and tasting the results. And speaking of webinars, let me just tell you a little bit more about the cycle that we are launching. So we have learned that IMTA and control systems can feel very complex. We have seen that this is a full nexus of fish, plants, microbes, water, feeds, and energy all working together. Also, these systems require serious CapEx investments. So getting the basics right is very important. It's very important to understand how everything connects before we jump into these processes. And this is exactly what this webinar cycle delivers. Our goal is to break down into pieces so that you can see the full picture in a more easy way. So coming up, we have microalgae engines turning wastewater into soil value. Here we will see how microalgae bioreactor captures fish waste nutrients and CO2, producing high protein spirulina that becomes feed or compost to regenerate your fields back on land. Also, we have a session dedicated to from science to plates. So algae and bioponic help us as let's see if they are really circular agriculture solutions. So here we address the full chain from lab tested algae strains through fish or plants co-production to real dishes that you can serve, showing that the business case from a bioreactor to the restaurant. So please, if you have not registered, the QR code is here on the screen, and it's now up to you to join on these game-changing sessions. Thank you. Again, theory is just a start, and nothing beats seeing Ampliaqua running live at our open days. So we have prepared open days at Ampliaqua, and they present this great opportunity for farmers, for the industry professionals, for academics to engage directly with this next generation solution. And I will turn very quickly how each group can participate. So farmers and producers. Ampliaqua is currently advancing biostimulants and biofertilizers focused mainly on abiotic stress. Abiotic stress on the work that Ampliaqua is delivering includes soils with high salinity, soils exposed to extreme temperature peaks, and those that are under hydric stress. And these biostimulants, biofertilizers that are being developed, they address these stresses individual or in a combination. Currently, we have a pilot with Tetrademus obliquus. It's a microalgae, and we know that this is a microalgae which is quite rich in amino acids, lipids, and has these characteristics of being a slow release nitrogen. So let me reinforce that these open days provide the chance to evaluate these solutions, and you can even initiate your own pilots in your own farm. For the industry, the developments that I've just mentioned, they also offer significant potential. So Ampliaqua is currently functionalizing compost in partnership with a major producer of winter and spring summer crops. And this functionalization is part of a major national initiative, which is the one of PRR. It's called the Tech for Green initiative, which is led by Ascenza Rovensa. So it's Ampliaqua expertise that enables this target customization of organic matter for very specific challenges. So please bring us your challenge to the open day, and let's start to collaborate in the development of these functionalized composts. And of course, not last but not least, academia. Ampliaqua also offers opportunities for academia. Ampliaqua is currently participating in some EU projects, of course, under controlled environment agriculture. There are projects running, for example, with Wageningen University. So also take your opportunity to join, and it's very relevant for those that are in research. Ampliaqua is also launching a Marie Curie researcher exchange alongside with BGI on full aquaculture value chain valorization. At Ampliaqua, the Great Amberjack project and this leads in terms of its biotechnology unit for regenerative aquaculture. So if you want to join Ampliaqua and BGI in applying biotechnology to recycle all the byproducts from the aquaculture process, please join us and be at the forefront of innovation. Just let me remind you that places are limited. We will welcome everyone, but we have a physical limitation. So if you are interested, register now. I will show you how you can do it. And we will be very pleased to receive you and to show you Ampliaqua live. So following our open days, we have more hands-on. We have set up a series of workshops that provide structured hands-on progressions for deeper implementation. This is targeted at farmers and producers and other agri-food sector professionals. And let me highlight that we have one workshop per semester focusing on innovative topics that blend practical education with sustainability and competitiveness. We blend theory with a practical format, transferring scientific and technological knowledge tailored to your specific needs. And of course, we award a certificate of participation. This is a formal recognition of your skills development. Of course, the sessions build directly from the open days discoveries, and they will enable participants to apply biostimulants, RAS techniques, and also circular biosolutions in your operation. And these workshops are immediately followed by our flagship RAS capacitation course that is designed for immediate implementation in recirculating aquaculture systems for those that are interested. We will send out detailed programs, dates, and registration information. Will be published very, very soon, so please stay tuned. Stay tuned to what's happening at Ampliaqua. Finally, complementing this, we have our fairs and show cooking events where solutions become tangible experience. And here we invite chefs, retailers, consumers at large, showcasing the commercial potential of our bioponic products like our basil, pesto sauce. So come along, experience cooking demonstrations that highlight these innovative products for chefs and entrepreneurs. And of course, we also look for collaborations with local value networks to promote these products, and also markets that are interested in these premium foods. And for you, particularly if you are very skeptical about taste, aroma, and shelf life of these products, I invite you, because this is your chance to try firsthand these products, prepare to get, something truly unique and quite new experience. You'll be surprised. So we have prepared everything for you. But because you're not at Ampliaqua at the moment, we have prepared a very short video for you to have a look and feel of Ampliaqua's operations. Sofia, please, can we see?
O Ampliaqua é uma unidade de investigação e desenvolvimento em escala pré-comercial dedicada à inovação e sustentabilidade em 3 áreas principais: aquacultura, bioponia e cultivo de microalgas. Este projeto nasceu da necessidade de criar, testar e validar soluções que promovam a circularidade entre diferentes setores produtivos. A nossa visita começa na sala de germinação. Aqui, controlamos a temperatura, a umidade e o fotoperíodo para garantir as condições ideais ao desenvolvimento das sementes. Utilizamos sementes biodinâmicas produzidas segundo os princípios da agricultura biodinâmica. Neste momento, focamos principalmente no cultivo de manjericão e rúcula, mas também produzimos salsa, coentros e tomilho. Cada espécie permanece na sala por um período diferente. O manjericão por cerca de 3 semanas e a rúcula apenas 1 semana, antes de serem transferidos para as torres biopónicas verticais. É também nesta sala que cultivamos microalgas de água doce. Esta espécie é autotrófica, ou seja, necessita de luz e nutrientes para realizar a divisão celular. De 7 em 7 dias, ou até 10, dependendo das condições, aumentamos gradualmente o volume da cultura. Começamos com 6 litros, passamos para 80 litros e, por fim, atingimos 5.000 litros. Estas microalgas apresentam um excelente perfil nutricional, tornando-se uma fonte natural de nutrientes para as plantas e ajudando a reduzir a dependência de fertilizantes sintéticos. Seguimos agora para a estufa, que tem uma área de 630 metros quadrados, onde está instalado o sistema recirculado da aquacultura. Este sistema utiliza várias etapas de filtração: mecânica, biológica e química, permitindo reutilizar praticamente toda a água. A renovação diária é inferior a 10%. Durante a alimentação e digestão dos peixes, são libertados compostos na água sob a forma de amónia, altamente tóxica para eles. No biofiltro, essa amónia é convertida em nitritos e depois em nitratos, através da ação de bactérias nitrificantes dos géneros Nitrosomonas e Nitrobacter. As biobolas presentes no biofiltro aumentam a área de superfície, facilitando a fixação destas bactérias. Os nitratos resultantes já não apresentam perigo para os peixes e são, na verdade, um recurso valioso para as plantas. Com tecnologia de extração seletiva de íons, utilizamos corrente elétrica para captar íons positivos e negativos, criando um concentrado nutritivo que é aproveitado na bioponia, reduzindo ainda mais a necessidade de fertilizantes de síntese. Na estufa, o setor biopónico é composto por 25 linhas com 11 torres verticais cada. Cada torre tem 8 andares e capacidade para 48 plantas, permitindo-nos atingir uma produção de cerca de 13.200 plantas por ciclo. A irrigação acontece pelo topo das torres. A solução nutritiva desce por gravidade, alimentando todas as plantas, e depois retorna ao depósito. O ciclo de irrigação é ajustado consoante a radiação e a temperatura da estufa. A argila expandida, na base de cada andar, ajuda a manter as raízes úmidas durante mais tempo. Para o controle natural de pragas, utilizamos várias abordagens. Plantamos espécies aromáticas como tomilho, sálcia e coentros. O tomilho, por exemplo, funciona como repelente de mosca branca. Aplicamos pulverizações de bacilos turgienses, óleo de laranja, bovéria ou outro biopesticida natural para proteção contra pragas e doenças. Utilizamos também armadilhas adesivas amarelas e azuis. As amarelas atraem mosca branca e afídeos, e as azuis são específicas para tripes, uma praga comum em estufas. Também na estufa, continuamos o processo de scale-up das microalgas. Os balões de 6 litros, vindos da sala de germinação, são usados para inocular fotobioreatores tubulares de 80 litros. Após 7 a 10 dias, parte dessa cultura é transferida para os raceways, onde atingimos os 5 ml finais. Aqui, produzimos cenedésmos oblíquos, uma microalga de água doce, utilizada como biofertilizante e bioestimulante natural, graças ao seu elevado teor de nutrientes e compostos bioativos, com benefícios diretos na saúde do solo. Mais recentemente, foi adquirido para as nossas instalações um bioreator à escala piloto. Este equipamento permite a propagação de micro-organismos em ambiente controlado. Parâmetros como a temperatura, o pH, o oxigênio e a agitação são controlados e monitorizados continuamente durante o seu funcionamento, garantindo condições ótimas de crescimento para os micro-organismos, assegurando um elevado rendimento final. O nosso método inovador de descontaminação, à base de peróxido de hidrogênio vaporizado, garante uma esterilização eficaz das cubas de fermentação, impedindo a contaminação com patógenos prejudiciais ao desenvolvimento da cultura e à saúde do trabalhador, garantindo, deste modo, o cumprimento dos procedimentos de biossegurança. A importância destes cultivos prende-se com a capacidade de propagar micro-organismos benéficos, como bacilos, tricoderma ou microalgas heterotróficas, que posteriormente podem ser aplicados nos terrenos agrícolas como biopesticidas ou bioestimulantes. Estes organismos atuam na supressão de pragas e doenças ao competirem com patógenos, produzirem compostos antimicrobianos e reforçarem a saúde dos solos e das plantas, contribuindo assim para a agricultura mais sustentável e com menor dependência de produtos químicos. Desta forma, o bioreator não só complementa a produção de microalgas, como também expande o leque de soluções biotecnológicas que o Ampliaco oferece, unindo inovação, sustentabilidade e eficiência em toda a cadeia produtiva agrícola.
Thank you. Also, thank you to Paulo Frias that guide us through this visit through Ampliaqua. He's our director at Ampliaqua, so you will for sure meet Paulo. So currently, we have covered the full Amplicentro journey. So we've gone through the nexus of understanding the topic through our webinars, our hands-on through open days and workshops, to end up with tasting the results. Of course, you will taste the results also at Ampliaqua, but also through our fair. So if you are interested and you want to join our open days, please use the QR code on your left. And here you'll register your interest and you'll have more information. If you want to continue to follow the activities of Ampliaqua, we have a dedicated newsletter, so stay ahead with updates and exclusive insights. Please use the QR code on your right. And of course, for me, I really would like to see us build this future of agriculture together. And I think it's your participation that makes things happen. We cannot do it alone. So register, and we'll be very pleased to meet at Ampliaqua. So I hope that you have enjoyed what we brought to you. You know, time flies. We don't have a lot of time, so I will continue. We still have our speakers with us. I think there is just one question here in the chat. Yes, let me just also remind you that all these sessions are going to be available in our website at Food for Sustainability website, so you can check everything and revisit this webinar and all the wonderful insights that we have covered so far. So, and also because our time is getting close to the end, and I know some of you are interested in getting your certificate, I think I can start by sharing with you the final assessment also. Let me just share, but I would like just to still hear from our speakers their views on the other side of the coin, okay, that we have briefly touched on our discussions. And this is actually a question for Kevin and Ioannis. And Kevin and Ioannis, you actually mentioned this throughout your presentation. You touch on some of the controversies of this system. So I would like you to build so that we are more careful about what we need, on your views, what we need to prepare for the future. But how do you see these IMTAs evolving to overcome some of the challenges, like some critics about the variable nutrient uptake rates? So the predator wildlife interactions, particularly when there are species, and this is more applied to offshore, of course, when there are species that escape. Also, the economic barriers for small-scale farmers, the lack of knowledge on the long-term data on the impact of benthic impact, or even the scalability of offshore setups. So how can we prepare for what now are some of the current controversies about IMTA systems?
Well, I think the major challenge of these systems is the kind of regulations that we have in Europe. For technology, we're going to find out a way, one way or another. But the problem is the regulations, because we have to persuade people that they make the regulations that these things do not pose any threat on biosafety, that they can be environmentally sound and proven, etc. And best of all, people they do not understand that if you make, let's say, an edible algae based on waste produced by the fish, this is quite safe for human use. So if you ask me, regulation is the major problem that we have here in Europe. If you check what is going on with IMTA in the Far East, then you can see where the future lies. They have vast facilities, offshore facilities, working with 3-4 different trophic levels, and even marine. This is on marine, but also fluvial systems. For instance, Vietnam in the Mekong Delta, they started some 2 decades ago, let's say 20, 30 years ago, where the aquaculture over there was nil. And they started working with earthen ponds where they had fish, and from fish they were having shellfish and mainly shrimp, and also seaweeds, etc., and also holothurians, whatever. And they make 9 billions today out of that. And these IMTA systems. So I think this is the main picture. We have to learn, and we have to adjust our monoculture systems to this kind of integrated multitrophic systems, that they are much better in biosafety. I mean, I've been for the last 10, 15 years talking with people that they are working with IMTA systems, and they haven't seen any biosafety problem. No disease outbreaks. On the contrary, they are contained, all these kind of things. And nowadays in the Mediterranean aquaculture, for instance, we are facing a lot of new threats that they come from diseases, like Tenacibaculus, like these new bacteria that they make, or the viruses like notavirus, etc. While in these more, let's say, natural systems, this kind of disease outbreaks, they are suppressed somehow, because these seaweeds, to name one, they are interfering with the quorum sensing of the bacteria, especially these pathogens, okay? And they are developing what we call quorum quenching abilities, molecules, that they are suppressing these kind of pathogens. And I think we need to put a lot of effort in this, because I think this is the real future.
Okay, thank you. That's an interesting perspective, and of course that's we all need to learn from each other, and those that are more advanced, we should also learn from them, not reinvent the wheel. It's also interesting the perspective that you brought about how the system actually regenerates itself. I think we could continue for a lot more. I think we've started to hear something. So, Ioannis, I also hope that you stay in touch with us, and you at some point you can also come and visit Ampliaqua. It would be very good. Of course, I also extend this to the
Gladly.
rest of the audience. Now you know how to do it. So I need to come to an end now. We've passed the hour, but I really would like to thank you for being here. You know what falls next on next Thursday. It has been a real pleasure to guide you through this webinar, and also a big thank you to our speakers, Jonas and Kevin, that had to leave earlier, and of course to the great team, Sophia and Dimitra, for your relentless support to making these webinars possible. Thank you so much, and I'll be back next week.